BACE Study Journal by Castida Mullally. Topics that I struggled with included STS page, ELISA testing, plasmids in biotech, plasmid transformation, and monoclonal antibodies. STS page is a protein assay that confused me due to its general similarities to gel electrophoresis, which I myself have done a couple times. It separates proteins based on their molecular weight in a vertical position. Proteins are unfolded in a negative charge. I was mostly confused between this and gel electrophoresis, so I'd actively compare them and ask myself questions relating to the either to explain myself the difference between the two. ELISA testing is a type of laboratory test that detects the presence of antigens, antibodies, proteins, and other molecules and substances, like that of blood, for an example. It can help detect and diagnose diseases a person or animal may have. I got confused with the several steps that this included, so I created a couple flashcards in chronological order explaining it. Plasmids in biotech. Plasmids are used in general therapy, vaccines, DNA cloning, and various other biotechnologies. However, I was mainly confused on how they are manipulated in a sense to prepare them for this usage, so I provided myself with several examples and explained them to myself over a period of time. Plasmid transformation. Plasmid transformation is a process that transfers foreign DNA into a bacterial cell, which is then expressed by the bacteria. However, I was mainly confused on its process and how the result of the expression is separated from the bacteria and is processed to be used by humans and technology. As a result, I looked at its steps extensively, writing them down and studying them over. Monoclonal antibodies. Monoclonal antibody production involves injecting an antigen, usually into a subject animal, and receiving back the B cells that produce the antibodies for that specific antigen. However, when I was first learning about this sort of production, I was confusing it with polyclonal antibody production, where I thought that it was specifically aimed for various amount of antibodies rather than just one specific one. As a result, I provided myself with flashcards that emphasize their differences that I studied them with.